Meiosis by Lab Group 3. Interphase is the first phase. During interphase, the uncondensed genetic material, called chromatin, is replicated. Because of this, the individual chromosomes are not visible as they have not been condensed yet. The centrosome is also replicated, resulting in two centrosomes. Centrosomes organize the spindle microtubules and are each made of two centrioles. Next comes prophase 1. It is the longest stage of meiosis. The nucleoli disperses as the nuclear envelope breaks down. The centrosomes move away from each other and the spindle begins to form. The chromosomes also condense. Each chromosome loosely pairs with its homologue, forming a homologous pair where the corresponding genes are next to each other. Then, the DNA of the two non-sister chromatids are broken at corresponding locations by proteins. The synaptonymal complex forms between the two chromatids, where the broken end of one chromatid is attached to the broken end of another chromatid. This is called crossing over, also known as synapsis. Once the synaptonymal complex disassembles, the points of crossing over are shown as chiasmata. Afterwards, the microtubules attach to the kinetochores of the homologous pairs. The homologous pairs then move to the metaphase plate of the cell. During metaphase 1, the homologous pairs have aligned at the metaphase plate. Within the homologous pair, the chromatids of one chromosome are attached to kinetochore microtubules from one pole, and the chromatids of the other chromosome is attached to the kinetochore microtubules from the other pole. Anaphase 1 follows metaphase 1, where the aligned homologous pairs are separated. Composed of microtubules, the spindle apparatus that is attached to the centromere of each homologous chromosome pulls the pairs apart to opposite poles, or two separate sides of the cell. This is in preparation for two new cells being formed in the next phases. Once the chromosomes have been moved to distinctive parts of the cell, telophase 1 occurs, as nuclei form around each group of the chromosomes and spindle fibers are broken. Each nuclei contains a haploid set of chromosomes, which will become the haploid set belonging to each of the two daughter cells formed in cytokinesis. In cytokinesis, a ring of protein filaments, known as the contractile ring, forms around the equator of the cell and pinches the cell's plasma membrane inwards, creating a cleavage furrow. In plant cells, however, the cell wall prevents the cleavage furrow from occurring, and instead, a cell plate forms to divide the cell in half, becoming two daughter cells. At the end of cytokinesis and before prophase 2, no DNA replication has occurred, leaving two haploid daughter cells. Now we enter into meiosis 2. In prophase 2, the nuclear envelope is broken down and the spindle apparatus forms in both daughter cells. Chromatin condenses into chromosomes, and as the centrosomes migrate to each pole, the newly reformed chromosomes line up along the center line of the cell, also known as the metaphase 2 plate, in preparation for the next stage. A continuation of prophase 2, the chromosomes, which were each composed of two chromatids associated with the centromere, are lined up at the metaphase plate as in mitosis. The kinetochores of sister chromatids are attached to the microtubules extending from opposite poles, preparing the sister chromatids for separation. Similar to anaphase and mitosis, the sister chromatids are separated and pulled to opposite poles. This is made possible due to the breakdown of proteins holding the sister chromatids together at the centromere. This ties into the law of independent assortment, which states that the two alleles for a particular gene segregates into independent gametes, separating from allele pairs of other genes. It is also helpful to keep in mind the law of segregation, which mentions that genes have alternative forms called alleles. Moving into telophase 2, a nuclei begins to form around the separated chromatids at opposite poles. The chromosome begins to decondense. Meiotic division of the parent cell results in 4 daughter cells, each containing a haploid or unduplicated set of chromosomes. The 4 daughter cells are genetically distinct from each other and their parent cell because of synapsis and crossing over. After meiosis, the 4 daughter haploid cells undergo further maturation or gametogenesis to become gametes utilized during fermentation. For males specifically, the process of spermatogenesis yields 4 sperm cells, while daughter cells in females undergo oogenesis, resulting in 1 single egg cell from the 4 daughter cells produced in meiosis. And that's it. Thanks for watching!